Endergonic reaction. What is that? ATP. What the? Enzyme, catalyst, blah, blah, blah. All right, you know what? I didn't learn any of this. I can't do it tonight. I need my sleep.
Hello.
Megan,
Yeah.
did you study for biology yet?
Not really.
Like, I don't understand what any of this means. Like, do you think it's even worth it for me to study, or should I just go to bed?
Honestly, that's up to you. I'm going to bed now, so.
Okay, then I'm just gonna go to bed too. My grade already sucks in my class anyway.
Alright, same.
Okay, bye.
Bye.
Summary's okay. Oh my god, I can't do this anymore. I'm falling asleep. So bright in here.
Wait, what are you doing here?
What? Why is Paul on top of me?
What are you guys doing?
Why is it so bright in here? What room am I in?
Whose house is Yo, calm down. I'm Mrs. Young, and I'm the teacher of your dreams.
Mrs. Young.
Yes. So, today we're going to be learning about glycolysis. This is to help you guys with your big biology test. So, does anyone know what glycolysis means?
Are we dreaming?
Yes, you're dreaming.
Wow.
Hence, teacher of your dreams.
Well, I was studying, but then I think I fell asleep.
I was studying, well, not really studying, then I just fell asleep.
I didn't fall asleep, I just gave up.
I never knew that I would be able to dream about something so detailed.
Okay, so do you guys know what glycolysis means?
Where did you get that dress?
Do you appear in everybody's dreams?
Okay, just let her teach or else you're going to fail tomorrow.
Alright, fine. Let's listen.
Okay, so glycolysis means the splitting
Oh,
of two sugars. So,
Okay.
you have one sugar here.
Is that what sugar looks like?
Yep.
Okay.
You're going to take it. Now you have two.
Is that his shoulders?
Yes. Okay, so does anyone know where this takes place? In a cell.
In
Cytoplasm. Yeah, in the cytoplasm. So, what happens when it happens in the cytoplasm? Do you know what's released?
Isn't it 2 ATP?
Not necessarily. Glucose and oxygen are first released into the bloodstream, and then 2 molecules of ATP, 2 molecules of pyruvic acid, and 2 molecules of energy-carrying electron molecules of NADH are produced and released.
So it occurs in the cytoplasm, and what's being released?
So now we're going to review the 10 steps of glycolysis. So in step 1, a phosphate group is transferred from ATP to glucose, which makes glucose 6-phosphate. Glucose 6-phosphate is more reactive than glucose, and the addition of the phosphate provides extra energy, and it also traps glucose inside the cell. Step 2, glucose 6-phosphate is transferred or converted to its isomer fructose 6-phosphate. Step 3, a phosphate group is transferred from ATP to fructose 6-phosphate, which produces fructose 1 and biphosphate 6. This step is catalyzed by the enzyme phosphofructokinase. which can be regulated to speed up or slow down the glycolysis pathway. In step 4 of glycolysis, fructose 1 and biphosphate 6 split to form two 3-carbon sugars, dihydroxyacetone phosphate, also known as DHAP, and glyceraldehyde 3-phosphate. These two are isomers of each other, but only glyceraldehyde 3-phosphate can continue through the next steps of glycolysis. In step 5, DHAP is converted into glyceraldehyde 3-phosphate, which is its isomer. The two molecules exist in equilibrium, but the equilibrium is strongly pulled downward, and all of the DHAP is eventually converted. In step 7, 1,3-biphosphoglycerate donates one of its phosphate groups to ADP, which makes a molecule of ATP and turning into 3-phosphoglycerate in the process. Step 8, 3-phosphoglycerate is converted to its isomer 2-phosphoglycerate, and step phosphoglycerate loses a molecule of water, which becomes phosphoenol pyruvate, which is also known as PEP. It's an unstable molecule. In step 10, PEP readily donates its phosphate group to ADP, which makes a second molecule of ATP. As it loses its phosphate, PEP is converted back to pyruvate, and which is the end product of glycolysis. Now we're going to talk about the difference between anaerobic respiration and fermentation. There are two types of fermentation. There's lactic acid fermentation and alcohol fermentation. The first type we're going to talk about is lactic acid fermentation. Lactic acid fermentation is the process by which our muscle cells deal with pyruvate during anaerobic respiration. If your muscles are ever sore, that is when lactic acid builds up. It's very common among bacteria, and that's why bacteria that is present in yogurt makes for such use. Under normal circumstances, the muscle cells make use of oxygen to carry out normal cellular respiration. But in the event where there is absence or lack of such, typically occurring during extreme physical exertions like exercise, then it will undergo lactic acid fermentation. So, the pyruvic acid becomes lactic acid in this type of fermentation. The lactic acid is responsible for making the muscles sore and a bit stiff, especially the day after engaging in exercise. Alcohol fermentation is a totally different thing. This type of fermentation is usually seen in yeast and bacteria cells. Unlike lactic acid, the end product, where lactic acid is the end product, alcohol fermentation, the end product is ethanol alcohol or carbon dioxide. And anaerobic respiration is a type of respiration where no oxygen is required. Fermentation can occur without oxygen. Okay, so next we're going to talk about obligate versus facultative anaerobes. Obligate anaerobes are organisms that survive in environments without oxygen, and they can die even if they're released into oxygen. An example of this is Clostridium tetani, which causes tetanus. Facultative anaerobes are versatile organisms. They can live with or without oxygen, but they grow best under conditions when oxygen is present. Okay, the next step in cellular respiration after glycolysis is pyruvate oxidation. It's the next step in the cycle in order to capture oxygen from ATP forms, but no ATP is actually produced directly through pyruvate oxidation. At the end of glycolysis, there are only two pyruvate molecules that have extractable energy, and the organelle in which pyruvate takes place is the mitochondrial matrix, or the mitochondria. Step 1, a pyruvate molecule has a piece of carboxyl molecule cut off from it so that it only has a 2-carbon molecule. Step 2, the 2 carbon molecules are oxidized, and therefore the oxidation loses positive charged electrons that become picked up by NAD and create NADH+. Step 3, the 2 carbon molecules are then attached to the coenzyme A, or CoA, an organic molecule that's obtained from vitamin B5 to create acetyl-CoA. Is the citric acid cycle. It's the central driver of cellular respiration. It takes what?
Are these two, is this within the same step?
Yes.
Two processes within the same step?
Yes, this is both, they're both step 2. Okay, so it takes place again in the mitochondrial matrix for eukaryotes, but in prokaryotes it occurs in the cytoplasm. And there's 8 major steps to the citric acid cycle. Step 1. In the first step of the citric acid cycle, acetyl-CoA joins with a 4-carbon molecule, oxaloacetate, which releases the CoA group and forms a 6-carbon molecule called citrate. In the second step, citrate is converted into its isomer, isocitrate. This is a two-step process which involves the removal and then the addition of a water molecule, which is why the citric acid cycle is sometimes described as having 9 steps rather than 8. In the third step, isocitrate is oxidized and releases a molecule of carbon dioxide, which leaves behind a 5-carbon molecule. During this step, NAD+ is reduced to form NADH. The enzyme catalyzing this step, isocitrate dehydrogenase, is important in regulating the speed of the citric acid cycle. In step 4, it's very similar to step 3. There's a 5-carbon sugar that's oxidized, which again reduces NADH to NAD+ to NADH and releases a molecule of CO2 in the process. The remaining 4-carbon molecule picks up coenzyme A, also known as CoA, which forms the unstable compound succinyl-CoA. In step 5 of the citric acid cycle, the CoA of succinyl-CoA is replaced by a phosphate group, which is then transferred to ADP to make ATP. In step 6 of the citric acid cycle, succinate is oxidized, forming another 4-carbon molecule called fumarate. In this reaction, 2 hydrogen atoms with their electrons are transferred to FAD, producing FADH squared. The enzyme that carries out this step is embedded in the inner membrane of the mitochondria. In step 7, water is added to the 4-carbon molecule fumarate, which converts it into another 4-carbon molecule called malate. In step 8, which is also known as the last step in the citric acid cycle, oxaloacetate, the starting 4-carbon compound, is regenerated by oxidation of malate. Another molecule of NADH+ is reduced to NADH in the process. The final step in cellular respiration is oxidative phosphorylation, and the electron transport chain and chemiosmosis. So what is oxidative phosphorylation? It's the final stage in cellular respiration, and it's made up of 2 essential components, chemiosmosis and the electron transport chain. Chemiosmosis is the movement of ions across their electrochemical gradient. Primarily, it moves down. Free energy from a series of reactions that make up the electron transport chain is used to move hydrogen ions across the membrane, which establishes an electrochemical gradient. Electron carriers, NADH and FADH, donate electrons to the electron transport chain. Uneven distributions of H+ ions spread across the membrane will establish concentration and electrical gradients. So, chemiosmosis is used to make 90% of ATP during aerobic glucose catabolism. The production of ATP using chemiosmosis is known as oxidative phosphorylation. It's also the method used in light reactions of photosynthesis, which harnesses the energy of sunlight in the process of photophosphorylation. The electron transport chain is one of the most complex and productive pathways of cellular respiration. It doesn't matter what kind of energy it obtains, sunlight or food, it has to be converted to ATP. ATP is stored in phosphate bonds and is used to get things done in the body. When ATP loses its charge, it goes back to being ADP, which is adenosine diphosphate. This conversion is done through cellular respiration. During aerobic respiration, the electron transport chain produces 34 out of 38 molecules of ATP. So where does the electron transport chain take place? In eukaryotes, it takes place in the mitochondria, or the chloroplasts, and in prokaryotes, it takes place in the plasma membrane, specifically the mesosomes. So how does it work? Think of it as 3 parts: oxidation-reduction reactions, hydrogen ion gradients, and the phosphorylation of ADP. So, oxidation-reduction reactions, electron carriers NAD+ and FADH, are reduced to form NADH and FADH squared, respectively. NADH and FADH squared are like rechargeable batteries that can accept and carry electrons and H+ ions, which can be used later for cellular respiration. These electron carriers will be oxidized and will transfer their electrons to other carriers which are dispersed around the membrane. In aerobic respiration, the electrons are passed from one carrier to the other in a series of oxidation-reduction reactions, and to the final acceptor, which is oxygen, that combines with a hydrogen, resulting in water, which is a metabolic waste product. The hydrogen ion gradient, energy from each electron is used to send an H+ ion through every carrier molecule from one side of the membrane to another, which creates a proton gradient, which allows for differences in concentrations between sides of the membrane. The phosphorylation of ADP. The H+ ions, which are located on the side of the membrane with more concentration, will flow back via the proton gradient and be put through an enzyme called ATP synthase. As each H+ ion moves back across the membrane, the enzyme ADP synthase phosphorylates or adds a phosphate to adenosine diphosphate, which makes ATP.
Oh my god, I understand what glycolysis is now. Glycolysis is the first step in cellular respiration. It consists of 10 steps. Before the 10 steps are performed, you have a 6-carbon sugar, and after the 10 steps are performed, it's broken down into 2 3-carbon pyruvates and 2 NADs, which are energy-carrying molecules.
I get it now. The Krebs cycle is the second step in cellular respiration. First, pyruvate is transported to the mitochondria and loses carbon dioxide to form a 2-carbon molecule. Then, chemical energy is released to form NAD, fat, and ATP. Yes!
Oh my god. So the third and final step of cellular respiration is oxidative phosphorylation, which consists of the electron transport chain and chemiosmosis. The electron transport chain consists of proteins and organic molecules. Electrons are then passed down from each member of the transport chain by redox reactions. The energy released is captured by proton gradients and then used to make ATP through a process of chemiosmosis. I finally understand this, thank God. Big idea number 1. The process of evolution drives the diversity and unity of life. Since cellular respiration is a natural occurring process in organisms, and because the origin of living systems is explained by natural processes, cellular respiration contributes to us as humans growing and developing, and other organisms as well, contributing to the big picture of evolution.
Big idea 2. Biological systems utilize energy and molecular building blocks to grow, reproduce, and to maintain homeostasis. Because all living systems need energy or ATP to live, this energy must be produced through a process called cellular respiration. In the process of cellular respiration, nutrients from sunlight or food are changed and or converted into adenosine triphosphate, which is also known as ATP. The 3 steps of cellular respiration are glycolysis, which only produces 2 molecules of ATP, the citric acid cycle, which again produces 2 molecules of ATP, and oxidative phosphorylation in the electron transport chain, which produces about 30 to 36 ATP, yielding the majority of the ATP which is needed to carry out everyday life activities like growing, reproducing, and maintaining homeostasis.
Big idea 3. Living systems store, retrieve, transmit, and respond to information essential to life processes. Your body must process if you need to undergo anaerobic or aerobic respiration, depending upon the amount of oxygen at hand. It must respond to these conditions to convert food or light into energy or ATP.
4. Biological systems interact, and these interactions possess very complex properties. Cellular respiration and photosynthesis interact because the products of one is what is needed to start the other. Cellular respiration is what creates energy and has very complex properties due to the fact that there are 2 main types of respiration, anaerobic respiration and aerobic respiration. Anaerobic respiration and aerobic respiration both convert glucose into ATP. They are very complex due to the fact that they have many, many steps involved in each process.